In order to maintain the proper functioning of our circulatory system, a balance has to be made between the coagulation and fibrinolysis cascades. Coagulation, or blood clot formation, occurs at a site of vascular injury to prevent blood loss while our cells are repaired. In contrast, fibrinolysis occurs to break down those blood clots and allow our blood to continuously flow. It is likely that you have heard of some sort of health condition related to imbalances in these two cascades. For example, if a blood clot is not properly formed, or if it is broken down prior to our cells being repaired, this can result in excessive blood loss, which is a characteristic of a disorder called hemophilia. On the other hand, if a blood clot is not broken down even after our cells have been repaired, this can lead to thrombotic events as seen in heart attacks and strokes. For my research project, I am studying a protein that is found in the plasma of our blood, which is called thrombin activatable fibrinolysis inhibitor, or TAFI for short. The activated form of this protein, TAFI-A, is a basic carboxypeptidase enzyme. This means that it is a protein that will cut other proteins ending in the amino acids lysine or arginine. TAFI-A plays a role in maintaining the balance between coagulation and fibrinolysis by slowing down the rate of the blood clot breakdown. Therefore, the study of TAFI is of great importance to the medical field. In order for us to understand how TAFI functions in our body, it is essential that we are able to study its activity when it's in its active TAFI-A form. There are current experimental methods used to study this activity, but each method possesses various downfalls. For example, the activity of TAFI-A cannot be monitored in real time, meaning you cannot monitor the activity as the enzyme is actually being activated. As well, very low concentrations of TAFI, which are similar to what would be found in our plasma, cannot be detected. Therefore, the focus of my research project is to design a novel fluorescence assay in order to monitor TAFI-A activity in real time with increased sensitivity compared to current methods. So far, I have obtained TAFI protein from baby hamster kidney cells and purified it. In my research, I will be activating TAFI to its TAFI-A form using an enzyme called thrombin with its cofactor thrombomodulin. This cofactor is used in order to increase the rate of the reaction. For my experimental assay, I have designed a substrate that TAFI-A will be acting on. This substrate is a peptide composed of 4 amino acids. In order, these amino acids are glycine, alanine, glycine, and arginine. A fluorescent molecule called tetramethylrhodamine, or TAMRA, is attached to the glycine end of this peptide chain. Another component of this assay is Evans blue dye, which is an anionic quencher. Since this quencher molecule is anionic, or negatively charged, it will interact with the positively charged arginine amino acid on the end of the peptide substrate. As a result, Evans blue dye will be brought in very close proximity to TAMRA, and it will quench TAMRA's fluorescence, or in other words, it will prevent TAMRA's fluorescence from being detected. The last component of my assay is the active TAFI-A enzyme. When this enzyme is added to the reaction mixture, it will cut arginine off of the end of that peptide chain. Loss of arginine means loss of the Evans blue quencher, which was associated with it. As a result, Evans blue will no longer be in close proximity to TAMRA, and it will no longer quench TAMRA's fluorescence. The resulting increase in fluorescence can be monitored over time in order to study the activity of the TAFI-A enzyme. There are many applications of this fluorescence assay. For example, we will be able to measure the TAFI-A concentration and activity within human plasma samples. As well, we will be able to study the activity of TAFI-A in blood clots in real time, and from this, we will be able to infer how TAFI-A can be regulated, especially in response to clot formation stimuli. There is also potential to identify individuals who are at risk for hematological or vascular diseases based on varying TAFI concentrations in their plasma and altered ability for them to form the active TAFI-A enzyme. My name is Kara Pico, and I hope to see you at my presentation so I can teach you more about my research and show you my results so far.